We've been talking about Mendelian genetics so far, and we've been looking at situations where you have a single gene that is governing a particular characteristic, and it has two different alleles, one that acts as a dominant allele and another that acts as a recessive allele. This is a fairly simple situation. In reality, of course, many different types of phenotypes or characteristics or traits are governed not by single gene situations with two alleles, one that's dominant and one that's recessive. In reality, many characteristics are the result of perhaps several different genes or a single gene that has several different alleles, and not all alleles exhibit simple dominant recessive relationships. So let's now explore some of those exceptions to the Mendelian rules, to Mendelian genetics, and we're going to start right away. Our discussion so far has really focused on Mendelian genetics. And for all of Mendel's genes, he was always looking at characteristics that were due to a single gene with two alleles for each gene, one allele being dominant and the other being recessive. His ratios in the monohybrid and dihybrid crosses were based on this simple relationship between alleles. However, not all inheritance patterns are this simple. A couple of exceptions are shown on this slide, and we're going to begin by talking about them, and then we'll explore a few more. The fun part of genetics is doing problems that involve these different patterns of inheritance. In most cases, you can use a Punnett square, as we've been doing for the monohybrid and dihybrid crosses. It's just that the interpretation of the genotype outcomes will differ. This lecture is primarily going to introduce to you the different patterns of inheritance. In some cases, I'll provide some problems that you can work on by stopping the video and figuring them out. There are also problems in your textbook and in Mastering Biology. I won't spend the time in this lecture taking up the answers to those problems, but I will provide them either during a synchronous session or by mounting another online video. The different inheritance patterns result from the nature of different gene products and what their role is in the cell. And I'll talk more about that in Friday's synchronous session. In Mendelian genetics, the heterozygote, shown in the middle of this slide, always shows the phenotype associated with the dominant allele. However, sometimes the heterozygote can show a different phenotype. Two examples of this are what is known as incomplete dominance and codominance. So let's begin with those. Incomplete dominance refers to situations where the heterozygote has a blended phenotype, which is really an intermediate between the two phenotypes seen in individuals that are homozygous for one allele or the other allele. An example is shown here, and this is a common example where we see incomplete dominance, is flower color. Here we have a dark pink or red flower. It's homozygous for the red allele. And we have a white flower, which is homozygous for the white allele. When you cross these two individuals, you generate offspring that are heterozygous, but their phenotype is different from both parents. You have some intermediate color, such as pink. Why don't you pause the video, do a Punnett square to work out the answer to this particular cross, and then choose the correct answer. Incomplete dominance is also seen in human traits or characteristics. A nice example of that is sickle cell anemia. We talked about sickle cell anemia in a previous lecture. You'll remember that it's due to a mutation in the beta hemoglobin gene. The result of this mutation is a phenotype where the red blood cells actually form a sickle shape, shown here. In most textbooks, it will tell you that the allele that causes sickle cell anemia is a recessive allele. However, that is not entirely true. In reality, the heterozygotes, individuals that have both a normal allele and a sickle cell allele, show evidence of incomplete dominance. Under normal oxygen levels, the phenotype is normal. Most of the red blood cells will look normal shaped. However, at low oxygen levels, there's a tendency for these red blood cells to sickle. In areas of high elevation where the oxygen levels are lower, they can become ill as a result of sickling in the red blood cells. Another situation where the heterozygote shows a phenotype which is different from the two homozygous phenotypes is in the case of something called codominance. In the case of codominant alleles, both alleles, if present in one individual, are fully expressed, meaning that both phenotypes that are visible in homozygotes are actually visible in the heterozygote. And the most common example of that, of codominance, is the human blood types, the ABO system. The ABO blood typing system in humans shows a number of differences in terms of things that affect patterns of inheritance. First of all, it's not a one gene, two allele situation. There are actually three alleles associated with this particular gene. And you can refer to those alleles as A, B, and O. The products of A and B alleles are codominant, meaning that if an individual is heterozygous and has both a copy of the A allele and a copy of the B allele, both of those will be expressed in the phenotype. The third allele, O, actually acts as a recessive allele to both A and B. This means if you have a heterozygote with an A allele and an O allele, the individual has type A blood. If you have an individual that has the B allele and the O allele, the individual has type B blood. It is only when an individual is homozygous for the O allele that you have someone who has type O blood. When working with the ABO blood type gene, we usually refer to the gene as I, and so you have two different alleles, A and B, where A is a superscript to the capital I. This indicates that they're both dominant alleles. And when they're both present, you have codominance. The recessive allele, which codes for O blood type, is shown as small I. Here we have the different combinations of alleles and the resulting blood types. This gene actually codes for an enzyme that adds a sugar to a carbohydrate on the cell surface. This results in three different antigens on the cell surface. There's the A antigen shown here, the B antigen shown here, and the O antigen shown here. I'll talk about this in a little bit more detail in my Friday synchronous session. Here's a nice little problem that uses the different blood types. So pause the video and see if you can work out the answer. Now, let's have some fun. This is a story about dogs. In our first cross, we start with a pure breeding black lab, and this male is mated with a female black lab who also comes from a line of pure breeding black labs. And when she has her litter, she has all black puppies. All right, here's our next cross. In this cross, we have a chocolate lab, brown lab, and he comes from a pure breeding line of brown dogs. He's crossed with a brown female who comes from a pure breeding line of brown, and lo and behold, they have a litter of brown puppies. Now one day, a breeder took one of the brown labs and crossed it with a black lab. And guess what? All the puppies were black. And then the breeder asked, "Hey, what happened to the brown fur here?" So he took some of these puppies and he crossed them. And guess what he got? A lot of the puppies were black, but some of them were brown. And the ratio of brown to black is shown here. So about 75% of the puppies were black and about 25% of the puppies were brown. Now, given this, you should be able to tell me which of these three scenarios or explanations is correct. If you have no idea, you really need to go back to the first video and start listening to these genetics lectures again. I'm hoping kind of that maybe you chose A. So when we get this 3-to-1 ratio in a hybrid cross, we're looking at simple dominant recessive relationship. And in this case, because most of the puppies are black, black must be the dominant allele. So probably we're looking at a 1 gene, 2 allele system for fur color. Now one day, a breeder took a black lab and crossed it with a brown lab. Now, what possible combinations or what possible outcomes in puppies might you expect to get? In this case, the outcome was some brown puppies, some black puppies, and some yellow puppies. Now, where did the yellow come from? This freaked the breeder out. So she went to a geneticist and said, "What the heck is going on here? I just crossed brown with black and I have some yellow puppies." Well, it turns out that in this black, brown, and yellow coloring system for fur in dogs, there's more than 1 gene involved. So more than 1 gene can affect a phenotype, and this is a really nice example of exactly that. And it's always fun to look at pictures of puppies. Here's the situation. The B gene that codes for black or brown fur color codes for an enzyme which makes pigment. The dominant B allele codes for an enzyme which makes a black pigment, and the recessive B allele codes for the same enzyme, but it's a slightly different version of the enzyme, which results in a brownish pigment. There's a second gene, however, which is known as the E gene, and this codes for a protein which is involved in pigment deposition. The dominant E allele codes for a protein which is required in order to ensure that pigment is actually deposited in the hair. The recessive E allele codes for a protein which is poorly functional or non-functional, and the result is that very little pigment can be deposited in the hair when individuals are homozygous for the recessive E allele. So when we look at this cute little yellow puppy, it's homozygous for the recessive E allele, and this means that very little pigment is getting into its hair, so it appears to have yellow or blonde hair. Now this puppy may have a genotype which codes for black fur or for brown fur, but it doesn't matter what that genotype says because it has a homozygous recessive genotype for the E allele. Most of that pigment, whether it's black or brown, is just not going to make it into the hair. This situation where the genotype for one gene can mask the expression of the genotype for another gene is often referred to as epistasis. So you may have run into epistasis in grade 11 or 12. It's also just a good example of a situation where you have more than one gene that actually affects the phenotype. And you can do really fun crosses with this type of system. And here's a cross where you have 2 parents, both of which are heterozygous, and you can figure out the outcome in the progeny here. So a nice exercise for you would be to try and do this cross without looking at these results, and then you can come back to this image and check your answer. The images of dogs in this little presentation come from Unsplash, and these are the photographers here. Now the situation we've just been talking about is a case where 2 different genes affect a particular phenotype, right? Which is the fur color of a dog. But there are lots of traits and characteristics both in humans and in all organisms that involve multiple genes. And to make it even more complicated, many genes are also influenced by the environment. So a nice example of this would be the height of people. If you measure the height of different adult humans, they can't be classified into discrete specific heights, meaning humans are not either 6 feet or 5 feet tall. They come in a huge range of different heights. And this is known as a continuous variation in the phenotype. This is a plot of the height of children, where the height has been plotted on the x-axis and the number of children measured is on the y-axis. And you can see this continuous variation in height. This usually reflects the fact that there are many genes involved in this particular phenotype. In the case of height, we also know that the environment plays an important role. Clearly, if a child is in an environment where they're not receiving proper nutrition or enough food, this will affect their height. Although they may have genetic information to be 6 feet tall as an adult, they may actually only reach 5'8" if they're not receiving proper nutrition during childhood. And finally, the last example where we have a type of inheritance pattern which is different from the inheritance patterns that Mendel saw when he was studying peas. And that is those traits or characteristics that are associated with sex chromosomes. We refer to that as sex-linked inheritance. So just as a refresher, humans have 23 pairs of chromosomes. 22 of those pairs of chromosomes, which we see here, are known as autosomes, and they're simply not sex chromosomes. Humans also have 2 sex chromosomes. The sex of humans is determined by whether the individual has 2 X chromosomes, in which case they're female, or an X and a Y chromosome, in which case they're male. All mammals have 2 sex chromosomes, the X and the Y. The inheritance patterns associated with genes that are found on the X or the Y chromosome is different from your standard Mendelian inheritance patterns. And here we see another image of the X chromosome with the Y chromosome beside it, and you can see that the X chromosome is much larger than the Y. Both of these chromosomes derive from the same ancestral chromosome. Millions of years ago, there was a time when the Y seems to have rapidly lost many of its genes, and some scientists estimate as much as 97% of its genes were lost millions of years ago. As a result, the X chromosome has far more genes on it than the Y chromosome. The genes on the X chromosome are known as X-linked genes, and the genes on the Y chromosome are known as Y-linked genes. And although the presence of these chromosomes indicate the sex of the individual, it doesn't mean that all of these genes on the sex chromosomes are associated with the development of sexual characteristics or the sex of the individual. In fact, most of the genes are not associated with that. Now, because the X and the Y chromosomes are different and contain different genes, you might be wondering, so what happens at meiosis? How do you make sure that in meiosis 1 of the sex chromosomes goes to each of the daughter cells? And it turns out that during meiosis, these 2 sex chromosomes do pair up and they act as homologous pairs. So they pair up together and they remain together until anaphase 1, when the X chromosome goes to one daughter cell and the Y chromosome goes to the other daughter cell. This, of course, is only happening in males, where you have a Y chromosome. If you were looking in the eggs of females, there would be 2 X chromosomes. They are perfectly homologous, and they will pair up just as the autosomes do. How do the X and Y pair up? There are parts of these 2 chromosomes which are homologous. And this actually is supportive evidence that they share the same ancestry. We call the regions on the X and the Y that are homologous the pseudo-autosomal regions. They're homologous just as the autosomes are. And you can see that these pseudo-autosomal regions or PAR regions are found at the ends of the chromosomes. These regions, because they're homologous, do pair up during meiosis. There is crossing over between them. This results in chiasmata that will hold the 2 chromosomes together until anaphase 1. So the Y chromosome seems to be necessary in order for an embryo to develop into a male. And one of the key sex-determining genes, or male-determining genes, has been identified on the Y chromosome. This is known as the SRY gene. And the SRY gene codes for a transcription factor that turns on genes in embryonic gonadal tissue, which will lead to the development of the testes. Once the testis has been differentiated, it starts to secrete male hormones or androgens, and the androgens in turn will result in secondary sexual characteristics that are male. When we talk about sex-linked inheritance, then, we're talking about traits that are determined by genes that are on a sex chromosome. The inheritance patterns for sex-linked genes is different from standard Mendelian genetics because they differ depending on whether we're dealing with males or females. In the case of X-linked genes, alleles can act as recessive or dominant, just as we see with autosomes. But for Y-linked genes, there's really no recessive or dominant situation because there's only one Y chromosome. So any gene or allele on the Y chromosome will be expressed in the male. Let's talk a little bit more about X-linked inheritance. A very well-known example of X-linked inheritance or an X-linked gene is the gene that's responsible for causing red-green color blindness. You don't have to remember all of these details about what causes red-green color blindness, but I thought it would be helpful to you to make the connection between a gene, an allele, and a protein. Opsins are proteins associated with light detection. Here is a picture of an opsin protein here. You can see that it's got tons and tons of alpha helices. It's actually a transmembrane protein. And in the heart of that protein, there is a pigment called retinal. And it's the pigment that actually absorbs the light. Different opsins can absorb different wavelengths of light. The absorption spectrum is shown here for different opsin proteins. There's an opsin in your rod cells which detect low-level light and is not able to detect or distinguish color. And the spectrum for that one is the broken line here. This is rhodopsin. The other three opsins are associated with detecting or absorbing different wavelengths of light. There's the L opsin, which can absorb peak wavelengths in the red zone, M opsin, which absorbs green light best, and S opsin, which absorbs blue light best. These three different opsins are expressed in your cone cells, and it's your cone cells that are associated with detecting different colors. Two of the three opsins that are involved in color detection are found on the X chromosome. Here's our X chromosome here. Here's where the L opsin is, and here's where the M opsin is. It's mutations in either the M or the L opsin gene which can result in red-green color blindness. S opsin is found on chromosome 7. These opsin alleles associated with red-green color blindness are X-linked because they're found on the X chromosome, and they're also recessive to the normal opsin allele. Individuals that have all three normal versions of the color detecting opsins are said to have trichromatic vision, and individuals that are lacking a normally functioning opsin are said to have dichromatic vision. When you're working with X-linked genetics problems, you have to track both the X and the Y chromosome. So you're keeping track of not just the alleles, but also on whether you're dealing with females or males. And so usually you put the allele for the gene you're dealing with as a superscript to the chromosome on which it is found. If you keep this notation when you're doing the Punnett squares and genetics problems, it makes things much simpler and it's much easier to interpret your results. Now, I think most of you have probably done these kinds of problems in grade 11 or 12, so here's a straightforward X-linked question for you. Pause the video, spend a bit of time, and see if you can work out the answer. If you're someone who has never done these kinds of problems before, you've worked through the textbook and you're still really not sure how to do them, by all means let me know and I'll be happy to answer this question either in a synchronous session or with a separate little video that I can post on our website. So do some genetics problems and have some fun.